Hello everybody and welcome again to the lecture. So, we are discussing module 4 and we have been talking about the DNA damages and DNA lesions that are formed due to exposure of our genomic DNA to the external and internal agents. So, and we have also seen how the lesions are responsible for creating the mutations in the genome, which in turn is responsible for uncontrolled cell divisions. We have started with the oxidative DNA lesions. We have seen the how under oxidative conditions such as in the presence of free radicals inside the cell, how the DNA nucleobases undergo damages, undergo different kinds of reactions that produces the DNA lesions such as 8-oxoguanine, 8-oxoadenine, FAPU-guanine and all this, the damages of the pyrimidine nucleobases also. And then we have started working with the UV induced DNA lesions, how ultraviolet radiations are actually destroying the DNA nucleobases and doing the photochemistry. So, we have started with the thymine dimer and we have seen how that produces different kinds of photochemical reactions. So, today let us continue with that and we will start with the cytosine dimers. So, this is your cytosine, and here is the sugar. We will take the dinucleotide. So, you have the phosphate, sugar, and this H2. So, this is our cytosine, P for phosphate and cytosine. Now, if it undergoes treatment with UV radiation, then what are the photochemistry that are going to happen? H nu, and of course, primarily we are talking about the UVC radiation along with other UV radiations that are primarily responsible for the photochemical reactions of the DNA nucleobases, UVC and UVB most of these cases. So, first of course, you have the pi bond here, you have the pi bond there. So, it will undergo 2 pi plus 2 pi cyclo addition, and you expect to get amine in H2 this. So, this is the cyclobutane ring. This is cyclobutane ring that is formed because of reaction of two double bonds, and we call it cytosine cyclobutane pyrimidine dimer. Cyclobutane pyrimidine dimer, in short we usually write as CPD. And now this compound can have different stereoisomers. The major one is of course the cis and syn. So, this is H, this is H, this is H, this is H. All H are up the plane, and therefore, The relation between this and this is cis, because they are both in the same up the plane, and relation between this and this are also the same, also the cis. So, this is termed as cis and this is syn, cis-syn, cytosine, cytosine, CPD. That is the major lesion that is formed, plus up the plane, this is up the plane, this can be down the plane, this can be down the plane. So, this is cis and anti, cis anti, CPD, plus this can be trans and syn, trans and syn. Trans and syn configuration would be something like this. H up the plane, this H would be down the plane. So, these are trans, this is up the plane. So, this is syn and this both have to be syn. This is same. So, this is also syn, this is syn, this is trans, this is trans. So, this is trans syn CPD lesion of cytosine, cytosine. Now, let us pick up this one and move forward, the major one. There is something else that are going to happen with that. In H2, double bond in sugar, phosphate. Let us stick to cis and syn. Now, this one does not stop here, because see you have the amine groups here, and once it loses the aromaticity. So, once you form the cyclobutane pyrimidine dimer, the double bond character is lost and therefore, the compound loses its aromaticity, and then this group is kind of isolated. The double bond is not delocalized over all through the ring. So, that makes the amine group quite susceptible for reaction. So, in physiological condition pH 7.4, this amine undergoes a rapid hydrolysis. We call it deamination. So, it reacts basically with water, and that will give you OH here, OH there, this cis-syn of course. Now, this process is quite fast process, it happens spontaneously in water. And then if you look there, this is basically a enolate form. So, it will undergo tautomerization to produce a ketone. This should be NH. This. Now, is this structure familiar to you? Can you recognize this? If you look here, so you have started with cytosine, which has the amine. Without the amine, with just the double bond oxygen, if you had the double bond here, this would have been uracil. So, this is now basically a uracil CpD. So, you have basically come down from a cytosine dinucleotide. After the photo reactions, you have finally have come down to a lesion, cyclobutane pyrimidine dimer lesion, not for cytosine, but for uracil. And in a short while, we will see how this is responsible for induction of mutations in the genome. And now, cytosine, the other photo products. What other kind of photochemistry that might happen? So, this is it. Now, this compound you can write in the enolate form this, the imine form, right, that is another tautomer of this form, sorry, it is a double bond here. And now, just like thymine, this can also undergo Paterno-Bucchi reaction between, if you have a double bond carbon plus, in this case this is carbon heteroatom, in this case this is NH. Under radiation, you can have the cycloaddition reaction, which is known as Paterno-Bucchi reaction, we have talked in the previous lecture. So, this kind of geometry that would be forming between the carbon and the heteroatom, that is why this has a different name. So, this is also a 2+2 cycloaddition reaction, 2 pi plus 2 pi cycloaddition. So, the same thing can happen here between these two, and that will generate here is the in, here this would be your I should write the oxygen intermediate first. This is formed, and you have the double bond here, and this is your double bond oxygen, correct. 1, 2, and then this should be an N. 3, 4, 5, yeah, 1, 2, 3, 4, 1, 2, 3, 4. All right, this should be NH. So, this intermediate for the thymine dimer, we have seen that this one was called the oxytane intermediate with the oxygen here, and with the nitrogen here, this is called azitidine intermediate. And of course, this is not stable. So, this rapidly will break down in this fashion. Here you have the hydrogen. So, this will come, this should be out, and what you will find is this. So, this ultimately comes down the plane. Here is your phosphate 1, 2, N. Now, this has become double bond over here, double bond, you had another double bond here, yeah, this is it. So, this is your 6,4 cytosine lesion or 6,4 photoproduct, and this can further undergo isomerization in presence of radiation, not in the same light, but with a little bit higher wavelength 320 nanometer around. This undergoes isomerization to give you the Dior valence isomer. This double bond comes here through the nitrogen, of course, and then this goes there. It is just isomerization. So, this happens under, it is a isomerization under 300, around 320 nanometer light. In H2 in here you are producing amine, and then if I write the Dior isomer, so this is the Dior isomer. Here you go. Dior valence 6,4 cytosine lesion, or you can also call it photo product. Okay. The 6,4 photo product or these both, the normal 6,4 in this position can undergo rapid deamination, as we have seen before. And that will produce ultimately one uracil kind of thing. Of course, this can also undergo, but this is to some extent stable because there is no double bond attached to it. I am writing the plain version, it can be the Dior isomer also. in here, here, or the dior, you can get the uracil. So, this would be, now you can call it, if it is the 6,4, I have written the 6,4 uracil P cytosine basically. Listen. So, you see how the structure looks very different compared to when you had started with the original dinucleotide. Now, the structure is very much different in this part, and therefore, it can pair up, or when it forms the base pairs, it does not always forms the base pair with the, we have started with cytosine. So, initially it was having the base pair with guanine. Now, after changing the structure so much, after alteration of the geometry, then and the property of this molecule, then it no longer pairs with the guanine, it pairs with some other nucleobase that we will see. Therefore, it will change your genomic sequence. Now, quickly, if you take the thymine-cytosines, so we have said that primarily when you are exposing the DNA to the UV radiation, primarily the pyrimidine nucleobases are susceptible for reactions or to do the photochemistry. That includes thymine, cytosine, and if it is RNA, then uracil. So, it can be thymine, it can be cytosine, or it can be thymine-cytosine or cytosine-thymine. So, if you have the thymine-cytosine, then what are the products that we will be getting? This is your thymine, this is your cytosine. So, TPC, and I am always doing from this when I am writing this fashion, this means the 5 prime and this is 3 prime. Okay. It can be other way around also, and you can write the relevant photochemistry or photo products. So, UV radiation. So, when we are doing the UV radiations, UVC radiation, as I have said, is the most intense or have the highest impact on the nucleobases, and that usually we do it at 254 nanometer. It can range of course from 250 to 280 nanometer. If you irradiate any light in between this, you will see this photochemistry. So, let us write only the products. So, you can have this followed by, of course, okay, I will do it right here. This will be formed initially, followed by hydrolysis or deamination. That will eventually give you uracil here. There is a thymine, I am sorry. Thymine. So, you get thymine P uracil CPD. I am not writing the stereochemistry, it majorly of course is the cis-syn, and then additionally you can have the trans-syn and the cis-trans, cis anti configurations. So, here again from the cytosine you are coming down to uracil plus the 6,4 here you will have the methyl group and the NH2, this sugar, phosphate, and here this is the 6,4. There is no question of hydrolysis here, and the corresponding dimer. Phosphate and double bond here. This is the dimer. So, these are the photo lesions or the photo products that you can expect if you irradiate a thymine-cytosine dinucleotide. For the sake of our simplicity, we have only taken the dinucleotide. They are actually part of the, these are actually part of the DNA. Now, let us see how these structures can induce mutations in the genome. So, for example, let us take 5 prime 3 prime, a CC sequence, opposite to it has GG, that is the normal sequence gene double helix 3 prime to 5 prime. Now, you have irradiated with the UV light, UV radiation, and let us say you have formed the CPD lesion here first. So, a dimer I am writing is as double bond C because it forms two bonds, right, one for the single, and then you have this is cyclobutane ring basically. You have GG here. Now, this had undergone deamination and it will give you uracil, that was what was produced. And now comes the important point, that when you go for further replication process with this, see in one case this will be divided into two strands through the replication fork of course, but it will be divided into two individual single strands even in the fork, and this should be acting as one template, and on top of it the new DNA would be synthesized by the set of those enzymes, replication. So, it will be fine. So, this is fine, you have your original DNA that is being replicated. Other case, you have now uracil as the template. Now, using this your new strand would be synthesized. I will show you little later that of course, that this thing would be repaired immediately, but many times they are overlooked by the repair systems and it stays like that. Then what is the problem that is going to happen? So, you have now the uracil and this was in 5 prime to 3 prime orientation. Now, uracil does not exist in DNA. In DNA what exists is thymine, right. So, the polymerase systems when this is using this as a template and synthesizing the complementary strand, they will initially read this as thymine because it is a pyrimidine, and they will take off adenine against it. So, the new strand, this is the new strand, will take adenine here, and then eventually when this goes for further replication, eventually this should be taken as template and this should be replaced adenine adenine, and you will have thymine thymine. So, eventually this damage would be spotted and would be relieved or would be executed, would be repaired. So, but in the meantime you have synthesized a template strand which is a pure DNA strand without any damage here. So, that would be taken as a proper template and it will take up thymine thymine against it and it will go on. So, what you get here 5 prime to 3 prime strand are thymine thymine. What did you have originally? Originally you have 5 prime to 3 prime strand, you had a cytosine cytosine. So, you are getting a double point mutation. So, cytosine cytosine into thymine thymine, double mutation or double point mutation. Single mutation is called point mutation. If there is only one base that is mutated, that is called point mutation, or also known as single nucleotide polymorphism, and this is called double point mutation or simply double mutation. So, that is how the DNA lesions dimer, particularly this dimer and cytosine cytosine dimer is responsible for inducing a double mutations, which is a big reason for creating the genomic malfunctions. So, in fact, this has been observed, the tumor suppressor gene, there is a name for it. I mean, one particular gene called P53 is a tumor suppressor gene, and in P53 this has been observed a cytosine cytosine sequence has been seen to be mutated to thymine thymine sequence, and that is of course is one of the reason for the malfunction of the P53 gene. And now let us look at the other lesion, 6,4 lesion. Okay. So, this is how the dimers are formed or the photochemistry goes on. 2 plus 2 cycloaddition, this is thymine, TP, T. I have written D also because to specify the deoxyribose, and then 2 plus 2 cycloaddition reaction, it undergoes cis-syn CPD, you can see here, and then trans-syn, this is trans-syn, it can have 2 isomers. Then you can have the Paterno-Bucchi reactions between this double bond and this double bond, that is producing a 6,4 PP is photo product, 6,4 PP lesion here that we have already seen, and upon further isomerization under 320 nanometer radiation, it will produce the Dewar valence product or Dewar photo product. Similarly, if you have the C dinucleotide, it will produce the majorly cis-syn, and then deamination will occur to produce the uracil cis-syn CPD. Paterno-Bucchi reaction, azitidine intermediate, this rearrangement followed by deamination. Rearrangement is this, as I have shown, this will come here, it will form the double bond and then it will break this carbon nitrogen bond. You will get the NH2 here, 6,4 PP lesion, and you had the deamination over here. So, you have the uracil moiety now, isomerization, that will give you the Dewar isomer. So, let us continue with the mutation. Five prime, 3 prime, A. This is your 5 prime. Now, 6,4 photo product. I am writing this fashion. this is 6,4 pp. You have AA. Now, it has been observed that once you form the 6,4 photo product, if you look back at the structures here, this is the thymine 6,4 photo product. You see that this is the new structure that has been generated, and this can pair up with a guanine. So, alternative base pair basically. T, when the prime strand is undergoing replication, then this should take a, I will write this as a new strand, plus when this strand is acting as the replication, it will give you the original DNA. So, you get your gene intact if this is your template. On the other hand, if this is your template, then you are having a G opposite to the thymine, and then as I have said that over time this should be gone away, but this will remain. So, you have the G, and here T, here ultimately a C will appear up. So, you had the thymine originally, now you are getting a cytosine here. So, it is thymine to cytosine point mutation that happens, and of course, majority of them will repair back. All right. So, these are the process how the chemistry is involved, or how you can understand the cause of mutations in the genomic DNA when they are exposed to either oxidative condition or the UV radiation. So, the structural parameters or the structural features play a major role in determining the mutations in the genome. Now, let us talk about the repair, how the cell is repairing, or what are the processes involved inside the cell that repairs the lesions or that repairs the DNA damages. So, repair of DNA lesions in cell. So, as I am mentioning again and again, our cellular system is very well protected, and we have really good mechanism that will find out immediately if there is a damage that is formed, and that we called there is a system called proofreading system, and then of course, proofreading systems will give the signals that there is some problem over here, and then the repair mechanism will go there and do the repair. So, there are mainly three kinds of repair processes that go on in the cell. One is called base excision repair. So, I am talking about both eukaryotes and prokaryotes. Base excision repair, in short, this is known as BER. Second process is nucleotide excision repair. In short, this is called NER. And the third one is mismatch repair. In short, mismatch repair, MMR. So, we will briefly see how these processes act. Before that, I will give you a statistics that how the mutation frequencies are actually happening in cell lines. I will give you a table. vector double strand DNA, and this is your leading strand, double strand and lagging strand. So, the data I have is for one particular type of cells known as cos 7 cells, or we call it cell lines. This is basically a cell that has been isolated from the fibroblast of, this is a kind of fibroblast cell, and this is been derived from kidney of monkey, that is in general the cos cell lines. Cos cell lines are fibroblast cell lines that has been obtained from the kidney of monkey. Cos 7 particularly is obtained from the kidney of one particular kind of monkey, that is in South African monkey. So, this is the cell line, and with that cell line people have studied that the frequency of mutations that are occurring within these cell lines. So, if you have, so that is why I have divided into two, one for the lagging strand mutation that can happen from the lagging strand using that as a template, or using the leading strand as the template. So, if you have without radiation, no UV radiation, no UV light, which means the normal DNA. The normal DNA, when it undergoes replication inside the cell, what is the probability of mutation? So, this table is for mutation frequency. In cos cell lines. COS cells. So, when you have the normal DNA and it undergoes replication, then also there are some mistakes that happen, but very less. The mutation frequency of the leading strand is really negligible, 0.01. Frequency means how many number of mistakes or how many number of mutations happening per unit time, maybe per second. It is really negligible, almost nothing. And if the lagging strand is used as the template, then it is slightly higher, 0.11, that is the mutation frequency. Now, if you induce 6,4-thymine lesion in the cell line, in the DNA of the cells, then for the leading strand your mutation frequency becomes 2.3, quite high, from almost a nil to quite a good probability or good amount of mutations that are occurring because of the 6,4-TT lesion. If the leading strand is acting as the template, if the lagging strand is acting as the template, then it is quite high, 4.70. So, high degree of mutations observed when the lagging strand is acting as the template, in which you have introduced a 6,4-TT lesion. If it is CPD, thymine, then it is less. Leading strand is 0.2, lagging strand is slightly high, 0.68, but still quite less. So, CPD lesions are not that much mutagenic, although they are formed in major quantity in the DNA when you expose it to UV light, but they are less mutagenic. On the other hand, 6,4 lesions and the dual valence lesions They are formed in minute quantity, their yield of formation is quite less, but their degree of mutation is very high. So, even a slight amount of formation of the 6,4 lesions can damage the DNA, can cause a huge amount of mutation in the genome, and that will change the function of the genome. Okay. So, this is basically, if you take what I have talked about is that is this, you have formed a replication fork 5 prime to 3 prime, and you are synthesizing the complementary. So, this is your leading strand as the template, and you are synthesizing the complementary strand. Then these are the mistakes that might happen. This is the frequency of the mutations in this strand that has been occurred, and similarly this is 3 prime to 5 prime end. So, this is your lagging strand, and you are synthesizing in a discrete fashion. Then what are the mutation frequencies in this newly developed strand are basically these. So, mutations will happen here. Okay. All right. I think we can discuss about the process of the repair in the next lecture. Thank you.